Despite novel therapies in AML, the overall survival still remains poor, and also novel T-cell-based immunotherapies, such, for example, CAR T-cells or bispecific, have shown limited efficacy. And now, recent studies have shown that the diverse genetic makeup of AML confers an intrinsic resistance to this bispecific T-cell treatments, and we specifically now hypothesize that the genetic aberrations of TP53 in AML confer an intrinsic but also an extrinsic resistance to these bispecific T-cell therapies. And we tested this hypothesis in a model using MV411, so an AML cell line with or without P53 knockdown, healthy donor T-cells, which is really important for our study, and the bispecific AMG 330. And we could indeed show that we saw a reduced specific lysis after co-culture, but to our surprise, we also saw a reduction in T-cell expansion and in inflammatory cytokine secretion. And we could also produce or reproduce these data using primary AML samples. So we then went on and asked ourselves if these results were secretome or surfaceome dependent, and continued by doing transfer assays. And here we could reproduce our results, so at least partially, but we then concluded that the results were at least partially secretome dependent. So we then went on and did an O-link analysis of the secretome after co-culture analysis. And we could find that lab TGF-β1 was highly produced in the knockdown supernatant, but not in the wild type. So we then focused on this candidate. So we looked at TGF-β1 in our co-culture setup and could find that this is highly secreted. And also when adding this now to the wild type samples, we found that the specific lysis, which was before higher in the wild type than in the knockdown, was then reduced equally to the knockdown samples. As we could still not completely explain the reduction in T-cell proliferation, we did then a RNA-seq analysis of T-cells after co-culture with the wild type and the knockdown samples. And we found that in the knockdown samples, the mitosis-related genes in T-cells were strongly downregulated, leading now or confirming this downregulation of T-cell proliferation. And now we are looking at mitosis in these T-cells and could also confirm in further in vitro assays that this one was strongly downregulated after co-culture with knockdown samples.